Hello, my name is Autumn Burlide, and today we're going to be discussing our proposal titled RNA Analysis via Whole Transcriptome Sequencing of Human Sebaceous Carcinoma Cell Lines. I'm joined by Caitlin McCarthy, Robert Mollenhauer, and Michael Hagler. To give a brief introduction to sebaceous carcinoma, it's a rare but highly aggressive cutaneous malignancy occurring in approximately 1 in 500,000 people. It typically presents as a pink or yellow nodule occurring on the head or neck, but most commonly located on the eyelid. Clinically, sebaceous carcinoma can be difficult to recognize due to its variable presentation, making accurate diagnosis a significant challenge. Histologically, sebaceous carcinoma may resemble benign lesions or other poorly differentiated adnexal tumors, complicating diagnostic interpretation even when biopsies are performed. As a result, patients are frequently misdiagnosed or experience delays in receiving appropriate treatment. Sebaceous carcinoma has a propensity to reoccur and metastasize, particularly if treated inadequately, making timely and accurate intervention critical. In the absence of a definitive early diagnosis, surgical management is further complicated by the challenge of complete tumor removal while minimizing cosmetic and functional damage, especially in the face and periocular region, where the wide excision may result in significant disfigurement to the patient. While histopathology remains the gold standard for diagnosing sebaceous carcinoma, distinguishing it from benign lesions or other cutaneous neoplasm remains a challenge. These limitations and complexities in properly diagnosing and treating patients have prompted a growing interest in molecular characterization of sebaceous carcinoma as a complementary approach for more accurate diagnoses and proper oncology care. In particular, early definitive detection through molecular characterization and quantification of gene expression can produce positive long-term outcomes for patients with sebaceous carcinoma. Moving beyond histology, early detection of definitive sebaceous carcinoma through molecular profiling of gene expression patterns has a potential to improve diagnostic accuracy and aid in personalized treatment strategies, ultimately leading to better long-term outcomes. Recent studies have identified recurrent somatic mutations in tumor protein p53, zinc finger protein 750, RB transcriptional corepressor 1, and NOTRapressor 1 as characteristic of sebaceous carcinoma but absent in benign sebaceous proliferations. Further investigation is warranted to characterize the molecular aberrations in sebaceous carcinoma more comprehensively, as they serve as potential biomarkers to enhance diagnosis beyond what histology can provide.
Our study has multiple interrelated objectives. Broadly, we are building off a previous study that cultured 3 Sebaceous cell lines. We will gather 10 patient-derived tumors and grow them to confluency. We will then confirm the presence of mutations in 4 genes using next-generation sequencing. We will then quantify those changes using whole transcriptome sequencing and perform further molecular characterization of Sebaceous tumors. Lastly, we will use a differential expression analysis to compare gene expression levels between a tumor group and a control group. For the control group, we will obtain tissue from healthy individuals. Our study will help inform targeted and strategic therapies of Sebaceous.
This workflow illustrates our strategy for comprehensive molecular analysis of sebaceous carcinoma. Following tumor excision, tissue samples are divided into 2 portions. One is processed for confirmatory histopathology to validate the Sebaceous diagnosis. The other is enzymatically disassociated and cultured into patient-derived cell lines for molecular analysis. From these fresh cultures, we'll extract high-quality RNA and DNA, enabling whole transcriptome sequencing and next-generation sequencing. Subsequent analysis identify differentially expressed genes, enriched pathways, and key mutational profiles. These molecular insights support earlier and more accurate diagnosis and uncover potential biomarkers and therapeutic targets, thus advancing both scientific understanding and patient care in SEBCA.
We can use molecular biology techniques to adequately identify sebaceous carcinoma, which is helpful to both the providers and the patients. SEBCA is common around the eye area and is easily confused with other diseases such as chalazion and conjunctivitis. A lack of proper and time-sensitive diagnoses can lead to a delay in treatment and therefore negative side effects, including the loss of eyelashes and distorted vision. Late diagnoses can also cause SEBCA to spread to the lymph nodes and parotid glands, reducing a patient's survival rate. By using an array of real patient samples and molecular characterization, we can simplify the concepts of SEBCA. We can use gene expression profiles and mutation signatures to allow patients to experience not only earlier, but more accurate diagnoses of this disease. Our research generates genomic databases and classifies various molecular subtypes, which can further the understanding of tumor biology in these underrepresented diseases, and therefore filling knowledge gaps found in cancer biology. Researchers can use whole transcriptome analysis to observe changes in cancer cells. When researchers understand these mechanisms, they can create targeted therapies and interventions with higher efficacy rates. As previously stated, SEBCA can be easily confused with other inflammatory diseases. Our study will not only promote early identification, but also more informed therapeutic decisions based on the atomic makeup of the patient's tumor. More personalized treatment can be achieved with molecular biology, which can increase not only awareness around this tumor, but can also improve patient survival rates. With a deeper understanding of SEBCA on a molecular level, researchers can compare this disease with other cancers and tumors, allowing for further comparative pathobiology research. These findings can assist in the development of various molecular diagnostic tools for SEBCA, such as cost-effective PCR-based tests. These tests could be standardized across healthcare settings, giving clinicians and various medical providers the ability to more accurately diagnose SEBCA.
Some concluding takeaways. Sebaceous carcinoma is frequently misidentified due to its overlapping clinical presentation and histological features with other lesions. By leveraging whole transcriptome sequencing on freshly cultured SEBCA cell lines, we offer more precise molecular insight than previously possible using FFPE-based analysis. This refined approach will allow us to do the following: uncover key mutational patterns and gene expression changes unique to SEBCA, facilitate earlier and more definitive diagnosis, and reveal novel biomarkers and therapeutic targets for future intervention. Our proposal sets the foundation for improved molecular diagnostics and personalized treatment strategies in this rare but aggressive malignancy. Thanks for listening.